Okay, terrific. So thank you everybody for being here. It's hard for me to do this without seeing you, but I'll do my best. So today I'm going to tell you about using X-rays to measure the solution structure of biomolecules, the present state of the art, and how XFELs can advance it. So I want to start by kind of parsing my title a little bit. So before I even tell you what I'm going to tell you, I just want to discuss the title. And there's a few things I want to highlight in here. The first is why biomolecules, and I'll have a slide about this in a few minutes. Then I'm going to talk about why solution structures, and finally why XFEL. So why biomolecules? That's because the commonly known biomolecules, which are proteins, DNA, and RNA, are critically important to life. And it's really the structure of these molecules that enables their interactions, and their interactions enable life. And in this talk, I'm going to restrict my comments to proteins. DNA and RNA are both very, very interesting biomolecules, but many of the studies that are ongoing at the XFELs are focusing on proteins, and so I thought I would start there because there's plenty to be learned. So as an overview to protein structures, I thought as an example it would be nice to look at the wide variety of structures that are present in nature. And the best place that I know how to do that is to just look at this poster from the Protein Data Bank, which shows really the wide variety of different structures that you can have. And it's just, you know, it's kind of awesome in you know, a great extent of the word. There's so many different structures. These molecules have different shapes, and it's really the shapes and the shape changes that allow these molecules to interact with each other. And that's a good part of how life happens. So then moving on to the second kind of keyword in the title, solution scattering. So why solution scattering? And much of what we know about the structures of biomolecules comes from the technique of X-ray crystallography. And I assume, you know, there have been a number of talks about crystallography in this series. It's a technique that's really, I think, getting a lot of new publicity, or there's a lot of new ways of looking at much smaller crystals, nanocrystals, using the XFEL. And crystallography is a fabulous technique, but it has its limits, and that's because the molecules are really constrained to be in a crystal. And so the parts of the molecule that you could be most interested in are the parts that are moving around or the parts that are flexible. And depending on the way the structure packs in the crystal, these pieces might actually be locked down. And so we get a lot of information from crystallography, but it's also important to realize that you're looking at a particular conformation of the protein. And I'll have an example later on in the talk that really drives that home. So solution scattering, on the other hand, can be applied to measure the structure of any soluble protein. So if the protein can be dissolved in a solution, it can be put into an X-ray beam, and its solution scattering can be measured. And one really nice thing is that solution scattering can be time-resolved, and so you can get dynamics, structural dynamics. And you can actually watch these molecules undergo dynamic conformational changes. You can see how the parts move and then how they interact. And by time-resolving these structures and changes, it's a wonderful opportunity to watch these molecules at work, which is something that I'm extremely interested in. So once we're talking about structural dynamics, a key question is here is what are the relevant timescales? So how quickly do the structures change? And these dynamics, as I've told you in the last few minutes, are just as important as the structures themselves. It's really the structural changes that these molecules have that allow them to interact, partner with each other, have complexes break up, and that's really important to how biology works. So many, many different timescales are biologically important. And the timescales of interest, you know, the timescale that you want to look at, really depend a lot on the parts of the structure that are moving. So I found this very kind of nice chart in a Nature paper from 2007 that basically spans timescales from the femtosecond out to the second and gives an idea of what the types of motions are that occur on these different timescales. And moving from the, the short time scale to the long time scale, it looks like there's perhaps smaller pieces. There's bond vibrations, there's rotations of different groups, methyl groups. The side chains rotate. As we move to longer and longer time scales, we like to think about having larger domains moving. So the picture that this kind of chart presents is that the smaller parts of the protein move on faster time scales, and as you go to longer and longer time scales, larger parts of the protein start to move. And this is very interesting, and I think one reason I'm making a point of putting this in here is later on in the talk, we might see some larger scale motions that happen on very short time scales that perhaps enhance our understanding of the picture that's presented by this simple chart. So keep this in mind. Okay. So ideally, if I ask somebody, what would be your ideal measurement, somebody who's interested in structural dynamics, I think the answer would be to be able to measure spatial resolutions of angstroms, which is really the size of individual molecules, all the way up through nanometers or tens of nanometers, which are the size of most, but not all proteins. Some protein and protein complexes in particular are much, much larger. But in general, angstroms through nanometers or tens of nanometers is a pretty good size scale for most isolated proteins. So that's the spatial resolution we'd like to have. It's really on the angstrom scale. And for time resolution, I think the chart on the last view graph made the point that it would be great to measure femtoseconds through minutes, so a very, very broad time scale. So now, because X-rays have angstrom wavelengths, they're really ideally matched to these length scales, right? So X-rays are, as I think everybody in the audience knows, it's light with a wavelength on the angstrom scale, and so light is very good at detecting objects that are larger than its wavelength. That's why X-rays are so effective at measuring on angstrom and larger length scales. So we've got that pretty much covered, and it's the time scale that's more challenging. So I'm going to, throughout this talk, one theme is going to be contrasting solution scattering measurements that are going on at present-day synchrotron sources with what's possible at the XFEL. So a really good question to ask is, what is it that limits the timescale for solution X-ray scattering measurements at conventional sources? And I would say that the signal strength is really critical here. And something that might not be obvious, but I think it's important to mention, is that proteins are not much more electron dense than the water that's around them, or the buffer that surrounds them in solution. And since X-ray scattering, and in particular solution scattering, depends on the electron density differences between your protein and the solution around it, those differences are small. And so proteins, as it turns out, are relatively weak scatterers of X-rays. And again, that's because we detect the electron density difference between the protein and the buffer around it. And so signal strength is, or it can be, a challenge, especially if you want to make very rapid measurements. So for this reason, you need a lot of X-rays to make the measurements. And there's a few different ways of making time-resolved measurements. One way is to do it in real time. And if you're thinking about the simplest possible explanation of a real-time measurement, that's just like taking a photograph with your camera, where the shortest exposure you have, it's the shortest exposure time of the camera, so you're taking snapshots in real time. And the limit on those kind of real-time measurements at traditional synchrotron sources is the pulse length. And the shortest X-ray pulse that's available is about 100 picoseconds. So there, if you're going to be making these snapshot measurements at a conventional source, you're limited to about 100 picoseconds. And that's not so easy to get to. But of course, there are ways to circumvent this, and I'll be talking about other strategies, for example, flow cells, that aren't real-time measurements. And we'll talk a lot more about that. But using these flow cells, specifically continuous flow cells, we can open up timescales that are greater than the microsecond timescale. So depending on whether you want to make a real-time measurement or whether you're happy using a flow cell, which kind of puts in a proxy for real time, you can adjust the time scale of your measurement to equal the time scale of the phenomenon that you're interested in measuring. So now the last keyword that I highlighted in the title, of course, is why we're all here, the new opportunities afforded by XFELs. And that's really the time structure of the beam. So the X-rays arrive in X-ray pulses that are about 40 femtoseconds long, the tens of femtoseconds long. These are at current generation sources. And most interestingly, the very, very high intensity of the beam really opens up new doors to us. And in this case, there can be about 10 to the 12th or more than 10 to the 12th X-rays present in each pulse. And just to set a scale for this, that's the amount of X-rays that we collect in about a second or a fraction of a second at a synchrotron source, like our local synchrotron here at Cornell, which is CHESS, the Cornell High Energy Synchrotron Source. So in 1 second at CHESS, we collect about 10 to the 12th X-rays, and we can get that in a single pulse at the XFEL. And so that's really remarkable and opens up new opportunities. So now, finally, I've arrived at the outline for my talk, and that I've parsed the title, I'll tell you what I'm going to tell you. So I'm going to talk about how solution scattering is extremely useful to look at structures and structural changes. And I've picked the examples of structural changes that are induced by light. There's many, many different ways to initiate structural changes, and I liked the idea of using light or photons because quite a bit of work has been done at conventional sources, and it's an area that's really growing in XFEL experiments right now. And also, light is a very, very important variable, as we'll see a little bit later in the talk. So again, I'm going to first talk about state-of-the-art synchrotron experiments that illustrate the value of this approach, why we can learn and what we have learned, which is a great deal about solutions and structures and structural changes. We've done that at synchrotrons, and then I will tell you kind of in the last third of the talk how XFELs have great potential to advance this field, and I'll briefly review experiments that have been carried out. And I should say that I have not been involved in the XFEL experiments on light-induced conformational changes, so for part of this talk, I'll be talking about my own work, which I'm very familiar with, and I'll also be briefly talking about the work of others. Okay, so just to really set the stage here, solution X-ray scattering. It's an incredibly simple technique. X-rays are incident in a very highly collimated beam that's coming from the left side over here. Use the pointer. They interact with the sample, and the sample scatters the X-rays, which means that the sample takes these initially very singly directed X-rays or photons and then spreads them out and scatters them through a wide variety of scattering angles. And we characterize the scattering angle by theta. So 2 theta is actually the scattering angle here. And then we collect this scattered radiation on a CCD detector, and we get a pattern that looks something like this. And the whole name of the game is figuring out what exactly got put in here to change the X-ray beam from being very, very collimated to this, widely scattered pattern. And so scattering is, kind of the quest for what it was that went in here, what is the shape, what are the different densities of the objects that are in here and their structures that converts this very collimated beam into this widely scattered beam. And so the data are often displayed as a function of a scattering angle here, theta, which is rewritten as Q, it's momentum transfer. But when theta is small, Q is about proportional to theta. So whenever I show you a plot that has Q on it, you can just read that as a theta scattering angle. Okay. And one thing that's very important to note here is that at conventional sources, this solution scattering profile is isotropic. So we call these pizza slices in my lab. When you mask out different bits of the scattering profile, you always get the same kind of intensities as a function of angle. There's really no difference in the structure as a function of kind of moving around the circle. They're kind of circularly symmetric. So here's a critical point that's often left out. which is background subtraction. So what we measure, remember I told you that we put this box that contains the sample into the beam, and the X-rays come in and scatter off of that box. But what's scattering is not just the molecules of interest. Those are shown by these red circles in here. But there's also scattering from any liquid that's around the molecules, scattering from any windows along the beam line, any walls of the sample cell that are containing the fluid here. And so we really measure the scattering from the object of interest plus a very large background. So it's important to subtract off the scattering from the background. So you need to create, in that box, a sample that's identical to the sample that you put in here. The only difference is that these red objects, the proteins themselves or the biomolecules, are missing. And you have to make sure that this is as identical as possible, because in many cases we're looking at small changes. And if you have even small mismatches, you can fool yourself into subtracting off something that's not the real background. So this background subtraction is really very, very critical. And you have to correct for any kind of changes in the intensity of the X-rays, any changes in the X-ray beam as well, in the times between when the scattering profile of this sample was acquired and the scattering profile of this sample. And only after you do all of that can you subtract the background from the first measurement, and then you get the scattering of the object of interest. Okay? So this seems like a trivial point. This is actually quite hard to do experimentally. It's not so easy. All right. So now that I've told you a little bit about that and we understand what we're measuring, now let's look at some solution scattering data. So this is background subtracted data. And here I'm showing you the computed scattering profiles of two different objects. One is a large and massive object, and one is a smaller object. And I'm just going to walk you through the scattering profile from low angle to high angle, and just tell you a little bit about, you know, what you can learn, what information there is in these profiles. And at the smallest angles, we call this small angle X-ray scattering, or SAX. So in the SAX regime, at the very, very lowest angles here, you can measure the y-intercept of your scattering profile. And it turns out that this y-intercept reports the molecular weight of your sample. And that's really neat, because if you have a sample that dimerizes with time, or things that dissociate with time, you will actually get a change in the forward scattering in the I0. And we use that a lot. It's very, very useful. You can get a lot of information that way. So as we move to larger angles, again, walking up the curve, the scatter into the lowest angles, so still near zero, but not at zero, gives you information about the largest spatial dimension in your sample, which we call the radius of gyration. That's just the mean square distance between all pairs of electrons in the sample. So you get the information about the largest spatial dimensions at the lowest angles. And as you go to larger and larger Q, or larger angle, you get information about smaller length scales in the problem. So, so Q, again, is kind of inversely proportional to a length scale. Large length scales here, small length scales here. And you can get low resolution shape information from this as well. And just to give you an example over here, you can, by measuring scattering out to a Q of about 0.3 inverse angstroms, which is a length scale of about 20 angstroms, you can get these rough envelopes, these low-resolution structural envelopes, which are the colored envelopes that are here, have the actual crystal structure docked into them. So from SAX, you can see the different shapes of these molecules, but again, you're looking at the envelopes here, the low-resolution structures, not the high-resolution PDB files that have been docked into them. But you can see that there's quite a bit of useful information that comes out of these curves, even scattering to a really relatively low Q, which is 0.3 inverse angstroms. You can definitely tell that these proteins have different structures. Some have holes in the middle, so there's a lot to be learned. So SAX has been very popular, And but recently there's a push to measuring at higher angles, which gives you information about smaller spatial dimensions in the sample. And this is a scattering profile. This is actually a profile of DNA, not protein. It doesn't matter that much for this talk, but I just want to point out that those past measurements stopped here at a Q of about 0.3. But if we go out to larger Q, there's really a lot more information. There's a lot of bumps and wiggles out here, and there's just a lot more information about what's going on smaller length scales in your structure. Okay. So clearly, from this part of the talk, I hope it's clear that structural information is pretty easy to get from SWACS data. That's what I call combinations of small and wide-angle X-ray scattering. But of course, you have to do a little bit of analysis to do this. So there are these reconstruction programs that allow you to rebuild these low-resolution envelopes. And again, there will be more examples of this that follow. Okay. So now I want to talk a little bit about time-dependent processes and structures and structural dynamics. And as we saw a few slides ago, this really depends on the time scale you're interested in and the biological questions you're asking. So there are a lot of biological questions. Many people are interested in how the protein moves, whether domains move, and you can imagine that would be expected to happen on the longer time scales because these are larger scale motions. Other scientists are interested in the side chain rotations. Maybe if a photon comes in and it starts a reaction, electrons are moved through the protein. There might be smaller, more subtle changes, and those tend to happen much faster. And so the time scale you're interested in is really coupled to the biology that you want to know about. So as I alluded to before, there's really two options: the real-time measurements or the use of a flow cell to convert position along a flowing chain into time. And both strategies work. They have different strengths and weaknesses, and they've been applied at synchrotron sources. And again, they can be readily extended to XFEL sources, and people, including my group, are working very hard on developing the technology to make this happen. But up next, what I'm going to do is give you an example of each of these two different types of measurements using a flow cell or a real-time measurement to answer a biological question. So my group has really specialized in using these continuous flow microfluidic devices to gain access to transient or dynamic states. And the way this works, it's really quite simple. We have some kind of a mixer, or just some kind of a microfluidic channel that where the molecules flow along it, and a reaction is initiated at some specific spatial location along the channel. This reaction can be the mixing in of a ligand. It can be illumination by light. You can really design anything you want in there, and then your sample flows along a very well-defined path, and then each location along this path corresponds to a fixed time after the reaction is initiated, and the kind of the proportionality constant is just how quickly the protein is flowing. So if you know how quickly your sample is flowing, you know where you're looking, you can tell what the time interval is between the excitation and your detection point. And what I really love about these continuous flow measurements is that you can expose for a very, very, very long time to get high quality data. So for example, if I'm interested in what happens 1 millisecond after a particular reaction has been initiated, I just move along the channel to the time point that's 1 millisecond downstream from the initiation point, and I sit and I train the X-rays on this one position, and I can measure for hours if I want. And I'm always measuring exactly what's happening 1 millisecond after the reaction was triggered. But it gives me a way to really boost the signal to noise. So as opposed to a real-time measurement where I have to take a snapshot that's a millisecond long, I can actually expose for as long as I want, but always be getting information about what's happening on the millisecond time scale. And again, this has been an extremely productive strategy in several different areas at a traditional synchrotron. And since I'm focusing on light-induced transitions, I'm going to tell you about some work that we've done that exploits this. All right, so now I'm moving to my first scientific example. Again, work from my group in collaboration with Cornell colleagues in Brian Crane's lab in chemistry. And this addresses the question of signal processing by proteins. So it's a very important question. How do proteins regulate the light response of organisms? So, you know, we live on a planet where if everything was fair, it would be light for 12 hours and dark for 12 hours, depending on where you live. That may or may not be true. But the point is that we have circadian rhythms that respond to light in the environment as one of several different stimuli. So the question is, how does a protein help you set your circadian clock? So what you need is a protein that transduces the absorption of a photon into some kind of an action or a conformational change. So the photon is the trigger, then the protein has to respond to that and then allow that change to be to affect other changes in a cell. So the particular class of proteins that we've been interested in are these love domain proteins, where LOV stands for light, oxygen, and voltage. These are a large family of proteins, and they're found in all areas of light. And they're signal transduction proteins. That's exactly what they do. They respond to changes in the environment. So, you know, when the lights come on, these proteins have some kind of a change that sets other processes into motion, maybe turns on or off other genes. So that's a lot of response, given that the stimulus could be as small as one photon. So the particular protein that we've been studying with the Crane group is called VIVID. It's a protein that's found in bread mold, and it's a light responsive protein. And this protein has several functions, and these have been assayed through knockout studies where they knock out this protein and see what happens. It's known that this protein regulates the production of orange pigment. So when you knock this protein out, that these mold become very orange, which is interesting to see. But the protein has also been implicated in circadian clock functions. It's known that it interacts with other clock proteins, and it affects the regulation, the expression of other genes. Okay. And the question is, how does this protein work? So for this work, we were very, very fortunate that the Crane group, they're mainly crystallographers, had solved the crystal structure of the protein. I'll point out for solution scattering experiments, crystal structures are useful, but they're really not an essential starting point, because as I showed you probably 10 view graphs ago, you can actually get low resolution structures. But I'm not going to complain if there's a crystal structure. So here is the Crane group's protein structure solved in the dark state. Turns out they could only get a crystal to grow when it was grown in the dark, so the protein was not light activated. So just to illustrate the difference between crystallography and small angle scattering, Brian Crane looks at this and sees the beautiful backbone and the alpha helices, but as a small angle scatterer, this is what I see, right? Again, it's low resolution, so there's a blob with a terminal helix. So in the interest of full disclosure, it's a dimer in the crystal, so this is what I measured, my dimer blob. So they were able to grow crystals of the protein in the dark state. It turns out the proteins like to be dimers in the dark state, and that's just the way that they packed into the crystal. But the light state crystal structure was elusive. Of course, they did what I would do, which is that you grow the crystal in the dark state, and then you turn the lights on. But the changes were very, very subtle, and they felt that the changes that they were seeing when they subjected the crystal to light couldn't quite explain the interactions that they knew that this protein had to have in vivo. And so they approached us and asked whether we could design a solution scattering experiment that would allow us to carry out this light activation for the proteins dissolved in solution. And so we had to overcome several challenges. The first is that we had to find a way to excite with light. So we want to have the proteins in the dark. We want to turn the lights on, zap it with a laser at a particular location in space, and then probe with X-rays at a fixed time afterward. And we want to be able to vary the delay time between when the light comes on and when the X-ray pulse is sensed. So to do that, we built a microfluidic cell that couples the X-rays and the lasers. And again, it goes back to this continuous flow design that I had talked about before. This is an incredibly simple design. It's a coaxial, two coaxial tubes. Looks like, you know, a gigantic paper towel roll, if you will. And if you look end on over here, you can see that there's fluid in the middle here. That probably should be colored this pink color, but that's the protein-containing fluid. It's surrounded by buffer on the outside, and that's actually quite useful because proteins are sticky, and the outer tube is made of glass, and the proteins just stick to the glass. And worse yet, they get seared into place by the X-ray beam. And so that changes the background, and it makes the background subtraction very difficult. So this kind of coaxial geometry is ideal for these kinds of experiments. So once we have a coaxial geometry, we have a laser beam that we direct at the flow right here. So the laser triggers the conformational change over here. The proteins flow down the channel for a specific length of time. And then we have a fixed X-ray beam that samples the molecular conformation at different times. And by moving the X-ray beam, by moving the laser beam, relative to the fixed X-ray beam, we can introduce a variable time delay. So this is how we can do a time-resolved measurement. Okay. So the experiment is extremely simple. We first just flow the protein in the dark, and we measure the structure. So we take solution scattering profiles, and then we turn the laser on, and we repeat the experiment. So now the light is on, the proteins can change their conformation, they flow down the channel, and we measure them, and then we repeat this experiment. And we can repeat this experiment with only the buffer flowing if we want to get good background subtraction. We have to do that. So we turn the protein off, and we repeat with only the buffer flowing to get the background subtraction so that we can then, you know, subtract the signal from the microfluidic device and from the buffer background. So here's the results of our first experiments right here. So this is the wild type vivid, so the wild type protein, and I'm now showing the scattering profiles as the log of I, because the important part here is that when we have measure the protein in the dark state, we get this darker curve, and when we measure in the light state, we see an increase in the, if you extrapolate this back, in the y-intercept. And that tells us that there's some kind of dimerization going on. And as I explained earlier in the talk, if you look at the y-intercept, and of course your concentrations are the same, and you've accounted for changes in beam intensity, you can see changes in the association state. This protein is dimerizing when the light comes on. And just to be sure that we're not damaging the protein, this isn't some crazy radiation damage effect, which you always have to worry about, we examined a mutant that doesn't respond to light the way that the wild type does. And in fact, there's absolutely no difference in the y-intercept here. And the protein concentrations are a little bit different between the mutant experiments and the wild type. That's why the signal is a little bit higher for the mutant experiments. But this convinced us that, in fact, the protein is actually dimerizing. So then we decided to start looking at structures to figure out what was going on here and to try to interpret our results. We looked at the dark state Sachs profile, and we tried to fit our scattering profiles, and it turns out that they didn't agree at all with the Sachs profile computed from the crystal structure. So first of all, remember that the crystal structure was a dimer that looked something like this. We got much better agreement when we took one of the monomers away. We just had a monomer in there. And what we did was we started with this crystal structure, and because we have the structure, we used some very sophisticated computational tools that would allow us to manipulate the crystal structure to see how we could get the best agreement between our scattering profile and a profile computed from the crystal structure. Okay? And this is the website, EMBL, the European Molecular Biology Lab, really has these wonderful Sachs analysis tools that are available for everybody to use. And so using one of these analysis packages, ensemble optimization method, what we found is that if we freed up, or we made flexible, this helix, the terminal helix, we could actually get the computed scattering profile from the model to look very, very much like the experimental scattering profile we acquired. And this just shows you that kind of the size of the changes. So this blue curve is the initial curve that we calculated based on the crystal structure. And as we allow more and more of that helix to become flexible, the curve changes shape quite significantly. And eventually we get this lavender curve, which provides a very, very nice, it goes on top of the data quite nicely. So what that told us then is that we could start building a very, very loose structural model. The crystal structure looks something like this, but what we're really seeing is flexibility of that terminal helix. So this bit, this alpha helix on the end, can really be moving around quite a bit. Okay. And then the next step is that once we had the information about the monomers, we could then learn about the structure of the dimer. What we wanted to do, we knew that the protein was monomeric in the dark state. When we added light, it turned into a dimer. So the next question is, what do these dimers look like? So here it's important to understand that we don't get full conversion to dimers. So we mocked up the experiment in the lab using other methods, other techniques, and we could measure the fraction of proteins that converted into the dimer state. Then the question becomes, can we extract the scattering profile of just the dimer if we know what the scattering profile of the monomer is and we know what the composition is? It's 30% monomer and 70% dimer, for example. And it turns out that we can, with a little work, we can extract the scattering profiles of the dimers. And this is, of course, assuming that there's just monomers and dimers present. And we get these scattering profiles. And once you have Sachs profiles that are background subtracted, you can then reconstruct the low-resolution structural envelope, which is kind of shown as this greenish low-resolution structure. And we've docked the crystal structure into it. And so the point is, of course, the crystal structure is absolutely correct. That's the way the proteins interact in the crystal. It's just that it's not the same as the solution structure. So in fact, this is a kind of a cartoon depiction. This is what we thought the dimer was going to look like, but in fact we're wrong, and it turns out it looks very much like this. And you'll notice there's a few changes. These helices are pointing up, they interact, and also there's contacts that are made along this kind of hydrophobic surface that's exposed. Once the light has an impact, the photon is absorbed, and side chains rotate, and this interface becomes a little bit more hydrophobic. We know that from other techniques. You can't easily measure that using small angle scattering. So what we learned is that the flexible region of the protein gets biased towards the thumbs up state following light activation, and then the protein dimerizes. And it's really nice that once this proposal was made from our small angle scattering measurement, once there's a specific idea, the Crane group went back and made certain mutants that actually favored the extended state, the thumbs up state in the dark state protein, and actually were able to capture this kind of thumbs up dimer. And they also made some other measurements that point out that the dimer is actually biologically relevant. So this was a really, really lovely use of solution scattering, and to me it illustrates how Sachs is useful, the kind of information you can get from it, the fact that we have a monomer to dimer transition, the fact that it's light activated. But an important point about this experiment is that we only captured the initial and final states because we answered the questions we were interested in. So our shortest time resolution was about 20 milliseconds, and that's just a result of, we were a little bit limited by the amount of protein that we had, so we had to flow it very slowly. And so when you have a finite beam size and you have a molecule flowing slowly through it, it's going to limit the time resolution of your measurement. If we wanted to, we could use a smaller beam, get more protein, and really fill in the points. But we answered the biological questions of interest, which is what is the light doing? and how is the protein transducing the absorption of a photon into a measurable event. And I would say that changing from a monomer to dimer is really quite a measurable event. Okay, so the take-home message so far, and again, this is from my group's work at CHESS at our local Cornell synchrotron source, is that both structure and changes are important. Biologically important changes can occur quickly. So by 20 milliseconds, this reaction was already done. And so these continuous flow measurements can also be applied for sub-millisecond timescales. We just, we got the answers we needed here on the tens of millisecond timescale. So now I'm going to move, for the rest of this talk, I'm going to be discussing work by other groups. So, and I'll start with synchrotron-based methods that probe light-activated changes on shorter timescales. So just another illustration of synchrotron methods. So this has been dubbed time-resolved wide-angle X-ray scattering, although I think they do, many of the changes occur at the wider angles. They're smaller structural changes, but if you look closely, there are really changes on the low Q region as well. And actually these experiments have been really beautifully reviewed, and there's a very nice reference down here on the bottom of current opinions piece. And again, carrying the theme of these light-activated processes, the way that these experiments are done is that differences are studied in curves that are acquired light on minus light off. And I want to point this out because there's no background subtraction required. So as opposed to our measurements, where we actually wanted the structure, we had to measure the structure. So measure something, subtract the background, get the scattering profile of the molecules, and then reconstruct that. But here, this is another way that solution scattering can be used. Here, structural differences are measured. So again, light on minus light off, and you subtract those and get the differences. And you really need to know what the starting structure is. So for these experiments to work, you have to know what the crystal structure of your starting state is, or have an NMR structure, or some other high-resolution structure. So in contrast to our continuous flow measurements, these are done in real time, and the X-ray pulse length sets the ultimate time resolution. So the experiments that I'm going to tell you about were done at the ESRF, and where the pulse length was about 100 picoseconds. And in these cases, a small volume of sample is used. So again, in contrast to our flow measurement, the sample is moved back and forth during an exposure to reduce the impact of radiation damage. So for these experiments, you don't need, buckets of proteins and many, large volume of protein. These are not continuous flow experiments. Okay. So the application that I'm going to tell you about is ligand release from hemoglobin. Okay, and the reference to this work is Nature Methods paper with the citation at the bottom here. And in this case, light is used to photolyze a bond, to break a bond. And the bond that's held in place is carbon monoxide, so the light comes in and breaks the bond. And the biological question of interest is, what is the response of the protein when the ligand is removed? And then, of course, the ligand rebinds, and what are the changes that happen after that? It's just that you can synchronize the ligand removal by using a laser to actually photolyze that bond. And for these experiments, because the time resolution is limited by the pulse length, changes are detected on a whole variety of timescales ranging from 100 nanoseconds, which would be really difficult to get in the continuous flow apparatus, but all the way up through 10 milliseconds. And the fastest events here on the kind of less than 150 nanosecond timescale really are tertiary structural changes on ligand release. So the protein itself does respond, kind of as a whole. And I thought it would be neat to look at how this experiment is done in a little bit of contrast with the other setup that I showed you. So this represents the X-ray at the synchrotron source. These are the pulses. They're 100 picoseconds long. They get chopped here. Pulses get selected, interact with the sample, and here a laser comes in from above. So these are real-time measurements where you're measuring for the pulse length, and then everything is collected on this detector. And again, the The signals before and after the photolysis are measured. The different signal is what's used here to interpret the structural changes for the protein. And you see here differences on a wide variety of hue or angles. So again, 0.3 and below is roughly the Sachs regime. This goes out very, very nicely into the wide-angle X-ray scattering region. And you can see that there's a lot of changes that happen here. Okay. So now, what if we want faster time resolution? Well, from that last example, it's clear that you can get down to about 100 picoseconds if you try really hard at conventional sources. But there's a lot that happens on shorter time scales, and from that chart of time scales, we could even guess at what some of those processes are going to be. So this is where XFEL comes in. So the XFEL pulses are short and powerful, as I told you in the beginning, and to date there have been two kind of solution scattering experiments on biomolecules that have been published, and the references are down here. And I'll very briefly tell you what each of them was, what the goal was, and what they found. Again, I was not involved in this work. I'm just reviewing what the current state of solution scattering is of these XFELs. Okay, so time-resolved SWACs, because it's really small and wide-angle X-ray scattering. And these experiments were carried out at LCLS, at SLAC, on the XFEL. So there's a couple of points. I want to highlight some differences between measurements of the XFEL and what we do at conventional sources. The first is that you have the real possibility of destroying your sample and maybe even your sample chamber by the XFEL pulses. So this requires a constant sample replenishment. And so hearkening back to my point about the background subtraction, if you're going to get the structure, you need to be able to have a reliable way to measure the background. If you're blowing up your sample chamber, it's going to be very hard. If you have a sample chamber, it's going to be very hard to exactly reproduce it. So background subtraction is a problem at XFELs, unless you attenuate the beam. If you have a much lower intensity beam and you don't destroy your sample, the sample chamber, you can get around this. So, but because many of the samples completely destroy or at least irreversibly modify the sample chamber, it's really easiest to detect structural changes and not the structures themselves. So you have light on versus light off, and you're not so worried about a structure minus a background. Although even so, you do have to make some assumptions that the background change is smaller than the changes that you're interested in to really reliably look at any kind of different signals. So for these measurements, beginning with structural changes, you need the dark structure, as in the hemoglobin experiments that I described to you in the last slide. And in this case, because the time scales are so short, molecular dynamics simulations are exactly a perfect match. They're on the right time scale to model the structural changes that you would measure. This is a great use of MD simulations. So very short time scales are accessible. So new information is also accessible. What is there to see? What kind of responses are there? What does the protein care about on these femtosecond timescales? So the first of the two time-resolved wax studies that I mentioned before really addresses this biological question. How is the photon energy absorbed by a protein without causing the protein to unfold? So that the proteins in the folded or unfolded state, they're not very different. Their stability is not very different. And even with a little bit of input energy, why doesn't the protein just unfold? So to answer this question, this group used multiphoton excitation of a photosynthetic reaction center. So they had a protein and they excited it, with many different photons. And the significant result is that they could measure that the protein dissipated this deposited energy through a large-scale, very rapid, single picosecond structural motion. So this measurement would be very, very difficult to do at a synchrotron source, where, again, the shortest time scale is about 100 picoseconds. Here, there's structural changes that are measured on the picosecond time scale. And again, just in the spirit of comparing experimental setups, here's the XFEL pulse coming in. Here's the light pulse coming in. Here is the injector that's continuously replenishing the samples, and in the diffract and destroy mode, the scattering profiles are collected on this detector. So again, the time scale for these measurements is no longer limited by the X-ray pulse length, okay, which is wonderful. Measurements can be made down to the single picosecond time scale. It's the delay time between the laser and the X-ray pulse that determines the time scale of the measurement. So this is not a continuous flow. There's actually, it's dialed in that the delay time, the laser pulse hits and then the X-ray pulse hits. And it's possible, and again, I took these very nice figures from the Nature Methods manuscript, and you can see that there's really two events here that are occurring on single to tens of picoseconds, and they are interpreted to be protein structural dynamics, which is very interesting because from my earlier plot, one might have believed that the structural dynamics needed microseconds to occur, but in fact there appear to be some concerted dynamics that are occurring on these very rapid time scales. And of course, words like protein quake, are in this manuscript to kind of describe the response. Okay. So that's the first one. And the second solution scattering study that's been carried out at XFELs uses lower intensity X-rays. Okay. And it's, in this case, photolysis is used to break a CO bond in myoglobin. So similar to the hemoglobin studies that were carried out at the traditional synchrotron source, here it's myoglobin, and you're looking at the photolysis of a bond. And it's very interesting that they saw similar single picosecond scale protein dynamics. And so just to kind of step back, to interpret their paper, and I'm summarizing their paper in a very short bullet point here. I encourage you to read both of these papers. But the suggestion is that rapid motions, these very rapid motions in proteins may be universal. And a very nice suggestion from this particular manuscript is that here the protein is responding to a very well-defined stimulus. And so the conformational change is induced by the arrival of a photon, very, very well-defined time. And that causes these rapid motions, but other motions are slow because perhaps they're statistical in nature and involve crossing barriers. But anyway, these two studies taken together really highlight the need to measure on all time scales. And then, so I hope that this work illustrates that XFELs have already begun to fill in the time gaps for these different measurements. So I want to close by just talking about a new topic, and I know that Delano gave a lecture to this group in the summer, and I expect that he covered much of this ground, so I'll keep this very brief to avoid repeating too much. But I want to point out that the measurements that I talked about are of structural changes, and they require a knowledge of the structure to interpret the changes. And so the question is, can XFELs be used to extract structures themselves? Kind of hearkening back to my first example where we were interested in the structure of the protein. And again, to get the structure, we need the scattering from the biomolecule alone in the background, and we have to subtract the background. And at a source like CHESS, this is easy to do. We know how to keep the background constant. We have an identical sample environment to get the background, and we average over many pulses. But at the XFELs, each pulse might be different. And remember, each pulse carries as many X-rays as we might have an entire X-ray exposure at CHESS. So there are other ways to get structures from solution scattering that are uniquely enabled by these XFELs. So again, and I expect you've heard about this in other lectures, so I'll be brief, but this is the simulated diffraction pattern from a single protein. And if we could measure the diffraction pattern of a single immobile protein, we could invert it to get the real space structure, and that's really the dream. Okay? And the very nature of the XFEL pulses allows a unique opportunity to do this. The pulse length is femtosecond, and the rotational time for molecules is much, much longer. And since each pulse contains enough X-rays to get a measurable diffraction pattern, each diffraction pattern reports particles that are frozen in space and time, and this is called snapshot SACs or snapshot diffraction patterns. And so here's an example. The beam comes in, the proteins are fixed during the exposure, and we get a snapshot diffraction pattern. And this is to be contrasted with the isotropic pattern that I told you about. This is kind of what we would measure at chest. These snapshot patterns are not isotropic. And it turns out that, in 1977, there was really a remarkable method proposed called the CAM method, where the diffraction pattern of a single protein can be retrieved by averaging over correlations or fluctuations in the diffraction patterns of many, many, many proteins, as long as they're frozen during the X-ray exposure. And here you can see that what has to happen is the intensity is correlated at different Q vectors, right? And so you compute these correlations as a function of angles. And here, the simulated diffraction pattern, this is from Delano's PhysRev letter. The reference is a few slides back. But here he's computed the simulated diffraction pattern of 10 randomly positioned and oriented proteins. And if you make tens of thousands of simulation, here 10,000 simulation, compute the fluctuations and follow the mathematical methods described in his paper, you can actually retrieve the single particle diffraction pattern from the average of all of these correlations. And this pattern is to be compared with the single particle diffraction pattern computed from a single protein. And you can see that there are some resolution challenges, but this is really remarkable. And I should say that this is a simulation, and because of background and noise, it can take many, many, many more than 10,000 patterns, probably more like 1 million patterns to do this in practice. But there are very encouraging preliminary results in non-biological systems. Here, one experiment on dumbbells that was taken at LCLS. This first reference and the second on silver nanoparticles. These correlations were measured at SSRL. And it's very exciting to think about, or to dream about, the applications to proteins in aqueous solution. It's still a ways off. What happens if there's no symmetry? Is this possible? 3D reconstructions. What about the solvent? And in fact, for anybody who's interested, Rick Kirian has a fabulous tutorial that I referenced here that really goes in depth into this. So to summarize, I hope I've convinced you that we've already learned a lot from solution scattering at synchrotrons. I think there's a lot more mileage in applying these methods, depending on the question you're interested in. The XFEL, as we all know, offers new opportunities for measuring solution scattering on very fast timescales and potentially new ways of measuring structures. And just stay tuned as we all ramp up, and really hope to, start making these measurements. I'm sure there's a lot to be learned. And finally, for the first part of my talk, my own group's work was the thesis work of Jessica Lamb, and these are the co-authors in my group, and then in Brian Crane's group in chemistry. And our funding was from the National Science Foundation through the Cornell Nanobiotechnology Center and also the National Institutes of Health. And then finally, I want to globally acknowledge everybody in the NSF STC and all of my BioXFEL colleagues and you guys who are listening to the talk. Thank you very much. And many of you can find yourselves in this picture. And of course, thanks to the NSF for supporting this wonderful effort. Okay, and that's my talk. Thank you for listening.